It's very nice to be here. And indeed, after coffee, some cake, it's time for food in the oral route. So let's see what we all consumed. As Vicky indicated, I think there are many types of particles, many types of applications, not only in food, but also in food production, in agriculture, so in the food production chain. It's indicated here. I'm not sure if you can read the small letters at the bottom, but what is said there is that in all the phases of food production, there are discussions on the use or really applications of nanotechnology, nanoparticles. And it ranges in various sorts. I think in the pesticides, people are discussing and developing liposome-like structures to encapsulate the active ingredient, to have targeted delivery in the insects or on the crops. And then on the other side, I think in the feed and food, the most well-known, well, it's always risky to call them nanomaterials, are the silicas and the titaniums. They have the E numbers, E5151. It's the silica and the titanium also. It's always questionable if those particles or powders really contain nanoparticles or not, but I think there's enough evidence and data to support that at least part of it is in the nano range. And this triggers discussions also in EFSA. Should we revisit the safety evaluations of these E numbers? Some of them are now currently being re-evaluated, and EFSA has launched a new task force or working group to look again at the implications of these kind of applications on their safety assessments. So in various phases of the food production, people use nanomaterials, and that's also as indicated by Vicky earlier, whole ranges of materials are being used and being studied. On the screen, the colors are not so visible, but on the other screens you see more of the colors, I think. But perhaps just the column is important for illustration. It ranges from all kinds of metals, gold, silver, to more the complex, inorganic, organic complex nanomaterials. So one would say that's all the evidence to look deeper into that, and also to try to measure them in food commodities and developing methods to assess the uptake of these particles. This is also, you can find many interesting pictures on describing the different properties of the nanoparticles. Metrics we've discussed earlier. And I think what is key and what's very special in nanotoxicology, if you want to study nanotoxicology, you need to have methods to measure these particles. And it's fairly easy to measure these particles in academic solutions or as a powder. It's more a challenge to measure them in real biological samples or in food, which is also in the end a biological sample. And then the question is, where should you target your method on? Which of these metrics are the most important? Or, if you are more pragmatic, and that's how we started, which are the easiest methods we can have or that we can think of to develop, and where can we then focus on? What's then, I think, striking or important to start with is, of course, the size. This is also related to the definition. The most common definitions define particles as being smaller than 100 nanometer. My background, I did not tell you that. My background is in a food safety lab. So we support the government in enforcing the food quality. And if we have legal definitions that nanoparticles are smaller than 100 nanometers, it's quite important to have methods, analytical methods, that can measure the size of these particles. Of course, what quite often then goes along is the shape, especially if you use EM techniques. But aspects like surface chemistry are then much more difficult to measure. So what I will do now, I think in my next couple of slides, is to show you a little bit more on the analytical methods that are being developed or that we have developed, and that I will use later on in my presentation to support the exposure conditions. I think you can roughly characterize the type of equipment used for the characterization of the particles in imaging tools, imaging of the particles themselves, or of cells that contain particles, and of course in previous presentations we've seen examples of that. Separation is quite crucial to separate particles from the biological environment, from the matrix, from the food matrix, or from the cell culture medium. And some of these abbreviations I will come back to later. And then of course after the separation you need to detect them, and especially for, well, not only because the first one, I was going to say especially for the metal particles you need ICP-MS-like techniques, but for the organic particles like the pesticides I mentioned, MALDI-TOF might be a very effective tool. I will try to tell you a little bit more about these methods in the coming slides. This is, I think, one of the working horses, I would say, now in nanotoxicology. I guess most of you will be familiar with normal ICP-MS, which you use to measure metals. And what is typical of a normal ICP-MS is the signal in the top graph. You measure the elements and you have a constant signal in your chromatogram, indicating that you have silver or gold or whichever elements you've selected in your ICP-MS measurements. You can change the software of just a conventional ICP-MS to change it in a sense that you measure only a very short intervals, and if you then dilute your sample enough, what you then in the end have is peaks of signal in your analyte, and these peaks correspond to nanoparticles. This is illustrated also here. In the ICP-MS, in the end, if you open it, you will have your detector with all kind of equipment before that, technical. But what it comes down to is that you have a detector, and if you dilute your sample and have very short intervals, at every measurement period, you will have that a plume of atoms corresponding to one particle enters the detector, and you get signals like this, and you can recalculate it, and then you in the end have a particle size distribution, a number-based particle size distribution. Especially this number-based is quite important in view of the definition of nanoparticles. The beauty of this method is that you dilute your sample. And in analytical chemistry, diluting a sample is quite easy, of course, and by diluting you also get rid of quite some of the interference of the matrix. So this is a method we quite often use in our experiments. It has, of course, its limitations. Depending on the material you are interested in, you have limits of detection, as always. For silver and gold, for example, we can go down to 10, 20 nanometer. Unfortunately, for silica, which is one of the mostly used materials in food, the detection limit is much higher. So I will come back to this method with some results later. The difficulty, or the nice thing is that, we assume that's inherent in the calculations of the size of these particles when measured with single particle ICP-MS. We assume a shape like this, that you will simply have a sphere. And this is of course not always the case, especially for silicas. You have a very more structure like this. And if you then measure this, if such a complex aggregate is entering the ICP-MS, due to the calculations, you assume a spherical shape, so that would underestimate the size of these particles. To try to understand this a little bit better, we not only use single particle ICP-MS, but we use it in combination with one of the separation techniques, and this is, in this case, it's hydrodynamic chromatography. or FFF, and this is an example or an illustration how FFF works. It's a separation technique, and it has two flows. It has a flow perpendicular, with the particles, and a flow just on top of that, and it comes down to that you separate the particles based on the size. And this is that in a flow. So what you in essence measure here is the hydrodynamic size of the materials. If you now couple this to the ICP-MS, you get a chromatogram from the with the size of the particle from the FFF or the HCC, and you get a signal from the single particle ICP-MS. You might recognize the typical pattern as I illustrated earlier for the single particle ICP-MS. And in this case, these are very initial results where we combine the FFF field flow fractionation with the single particle ICP-MS. We use gold nanoparticles, and gold are of course nicely spherical, so it's not a surprise that the two peaks of the abundance of the FFF and the single particles nicely overlap. But if you would have used silica, then you would have seen that the two lines are not so nicely overlapping as in this case. It's also a very interesting method if you have more complex materials. Not only one element, but combinations of different elements, which I think are in the next generation of nanoparticles to be expected. And then you get more interesting information on really what kind of particles or materials are you studying. And the last technique I'm going to highlight is one of the methods to show or to measure the organic nanoparticles. I think especially in food, when you want to deliver an active ingredient, a vitamin which is poorly soluble, or iron which has a poor taste in food, you want to protect it from the harsh conditions in the intestinal tract, to have a delivery in the intestine. And then the question could be, what is the risk of these nanoparticles? I think the traditional hardcore nanotoxicologists would say, well, the main risk of toxicology would be the metals or the metal oxides, and not necessarily these organic particles. But I think one needs also to consider the possible increased bioavailability of these particles. And then it's not necessarily a nano problem, but the delivery, so the kinetics, might be different. So then there are two ways to approach the detection of these capsules. Is either to measure simply the active ingredient, which can be fairly easy, of course. It's just a chemical, and you can use all kind of MS techniques to measure the active ingredients, or to try to measure the composition of the shell, which is not always easy, because especially if you go to the food domain, there are quite strict regulations on which types of lipids you can use. It's different from the medicinal field, I think, where you are more flexible. But in the food domain, well, you need to have food grade lipids, which also are frequently found just in the matrix. What we use there is again a combination of a separation technique, either ATC or FFF, to separate the particles from the matrix, and then with a MALDI-TOF, which is explained a little bit here, try to analyze the fragments of these particles, and then you can analyze the composition of these particles and know a little bit more if there's really if there are really organic particles in the matrix. And then you get this kind of information where you can show that you have the different components of the lipids present in your sample, and by focusing at the different masses, you can of course try to identify the composition of your capsules. not so much toxicology. I'm afraid I will not touch too much on toxicology in my presentation. As I said earlier, I think that the methods are quite crucial for nanoparticles to really show that you have particles in your exposure and that you are studying nanotoxicology. My topic is more on the oral exposure route, and of course I've showed you which particles and which types of particles we are exposed to. But the gastrointestinal tract, I think, is composed of several barriers. And we try to develop models for the barriers. And I think the first barrier, or the most harsh barrier, is of course the stomach. The stomach with a high ionic strength, low pH. I think, but this does quite a lot to the appearance of the particles. And the question could be, what happens to these particles, and does the history of where the particles have been in the gut or in the intestine affect the exposure? So we developed models for this. I think 3 systems. The first 2 are classical systems, although the biology on the Transwell system was also indicated by Barbara. For the lung model, you can have different models, simple models, which are very useful for screening studies. But if you want to have more biological relevant outcomes, you need to start with more complex models. And in our case, we frequently use Coatsome cells, which are traditionally used cell line in the pharmaceutical field. And we co-culture that with goblet cells to have a mucus layer on top of these cells. This is not only more physiologically relevant, but also really needed if you want to couple an epithelial model to a digestion model. This digestion model, it's really basic chemistry. We have different tubes in our lab, quite large tubes, that we spike with saliva and subsequently spike with the gastric juices, modify the pH depending on really the conditions we want to simulate. Do we want to simulate a fasted model or simulate a fat model? And then slight differences in the pH have been observed in humans. So the conditions are human inspired, but the tubing, well, of course, is far from human inspired. The beauty of this is that it's so simple. You have different tubes on your bench, and you can take samples when you want. You can make time series and then use the analytical methods that I indicated earlier to study the appearance and fate of the particles. And in my last slides, I will go a little bit more back to my title and show you the directions we are going into with microfluidic models. We use this in vitro digestion model with the tubes to study the fate of silver nanoparticles. This has been published a couple of years ago. And what you see here, it's quite interesting. Well, we thought it was quite interesting when we saw it. We characterize the particles in water, and then you have a nice size distribution. The same size distribution comes back when you have it in saliva. And interestingly, and if you then look at the gastric juice, so the condition, really the conditions in the stomach, well, the size distribution did not seem to be affected that much, but a lot of particles were gone. And this, of course, we could not explain in first case. And then interestingly, if you then move the same sample where we've taken one subsample from the analysis, move that to the intestinal phase, again, the size distribution much resembles the size distribution in the saliva. What in the end turned out is that we had to use a filter before the single particleized species, and that the particles have grown substantially due to aggregation in the gastric phase, but they simply did not enter the injection machine. And strikingly, and this is always puzzling, I think, in nanotoxicology, if we exposed this system to simply silver ions, we also in the intestinal phase found some nanoparticles. In nanotoxicology, it's always a discussion: how new is it? Some people argue we have been exposed to particles during evolution, so our body can cope with it. The counter argument could be, well, that's the natural background. Perhaps if you make them or introduce them into your food, you have different properties, different materials, and higher levels. But this also indicates that if you have silver ions, that, well, endogenously, some particles are being formed. So it's good to put these kind of things in perspective, I think. These are some EM pictures really showing the particles. If you would not have believed the graphs from the single particle ICP-MS. Here are some silver particles in the saliva matrix. You see a flowery structure here, which is just the background of the complex composition in the intestine. And here, so the white spots you need to focus on. These are the silver particles. And this is an example of a sample from the gastric phase. The black holes are the grids, and here you have a cluster of silver particles really glued together. And this glue, we did not look into this in this study, but later on I will show you a picture in a minute. This glue very likely is chloride. And then if you continue your sample, you again have the free particles, but that's not too surprising if you know that they are simply glued together due to the changes in electrostatic repulsion and van der Waals forces. We've seen the same for silica. We've used here the food grade silica, and that is a complex structure, so it's difficult to show you really how it works in model situations. But we used here HCC ICP-MS in the 3 stages of this in vitro digestion model to look at the presence and the size of these silicons. And again, as I showed you for the silver, it seems to be a common pattern. In the mouth we find nanoparticles, in the stomach only a fragment of that, and again a reappearance in the intestine, in the gut. When we look at this with model particles, again silica, but nicely spherical particles. The white spheres are the silica, the black holes again the grids. You see clusters of particles in the stomach conditions, and again a release of the particles in the intestinal phase. And here we looked a little bit more with SAM-EDX into these into the stomach, I have to say, stomach samples. The red is the silica and blue is the chloride, and for this reason I postulated that for in the silver study also the glue is likely to be composed of chloride. So really showing the dynamics in the intestinal models, and likely also in our intestine. So this is basic chemistry. It's becoming more interesting, at least I find it more interesting, if you now couple these models. So we coupled these models offline. We first incubated particles, and for the sake of detection we used in this case polystyrene particles. We digested the particles in this in vitro digestion model and subsequently exposed a co-culture model to the output of the digestion model, and now the mucus layer is becoming crucial, because in this intestinal phase we have bile acids. These are important components of the digestion, of course, but these bile acids are toxic to cells. So if we would not have a mucus layer on top of it, we would need to dilute our samples so drastically that the relevance of the study would be completely gone. And this is a top view image. The blue is showing a little bit of patchy structure, appearance of the mucus on the cells. What's now really interesting is that if you compare the dark green with the light green particles, the uptake of digested polystyrene particles seems to be increased compared to the non-digested, so the pristine particles. So that's the comparison of the digested and the undigested particles. What it also shows is that there is a huge dependence of the surface modification of these particles. The charge is different of these particles, and the charge of the particles affects the uptake substantially. What I'm not showing here is that this likely is due to the difference in protein corona on these particles. And if you digest it, you also digest the corona on the particles, which affects the uptake in the end. As this is toxicology, I have a few slides on toxicology. This is a study where we exposed CACO2 cells to silver nanoparticles, and it will address a key issue, I think, in nanotoxicology. This is simply a traditional experiment where we differentiated CACO2 cells and exposed them to pristine silver particles. And we used a confocal microscopy to see if the particles have been taken up by the cells. This again shows one of the limitations of visualization of particles, because only here, I think, for you in the audience, it's clear that the green is a reflection of the silver nanoparticles. That you see silver close to the cells. This blue is the nuclei, and very vaguely you can see the outline of the cells. So the silver is really in the cells, and this is a cross section through the cell that it's showing that it's not only on top, on the outside of the cells, but really in the cells. With some imagination, you could also see it here. These particles are 100 nanometers, so you have quite a strong reflection of these particles. But the smaller the particles, of course, the less of the laser light is reflected. So here we can also show that we have some silver nanoparticles in the cells. But for the smaller particles, it's getting quite difficult to convincingly show with this technique that the silver is in the cell, that the nanoparticles have been taken up. We are sure that it's there because we also combined it with ICP-MS. We did exposures, so the different sizes of silver nanoparticles, but also to silver ions, and looked at effects on gene expression. Well, these graphs are quite easily complex to interpret. These are the traditional heat maps you might be familiar with. And you see that we use different cells, so I will focus now on the CACO cells only. And you see that we have silver and particles. And what was striking, if you look at the heat maps, that the gene expression did not seem to be too different between the silver ions and the silver nanoparticles, which is also shown in a different way here. The different particles, sizes, and the ions in a Venn diagram. Trying to identify nano-specific effects. And it's easily seen that in this overlay, where we have all the sizes of the particles, that we found 130 genes that are affected, in this case upregulated by the exposure to nanoparticles. So if you would not have co-exposed your cells to silver ions, you would have said, well, that's a nice effect of nanoparticles. But it's striking that if you do only the exposure to silver ions, that we have 70 genes also affected by the exposure. Well, that might be a difference then, of course. You have 60 genes more expressed due to the silver nanoparticle exposure. But if you continue the analysis and more do the biological interpretation of the affected genes, the pathways affected in both types of exposure is really comparable. What's different is the kinetics, and that we are convinced, and we're now putting this down in a manuscript, that the same biological pathways are affected in the nanoparticle exposure and the silver ions exposure. And this is of course not too surprising, knowing that silver particles continuously dissolve, not only in medium but also in the cells. So that the ultimate toxicological agent, as it's called, I think, is simply silver iron. So you need to be very careful in how you design your study and how you interpret your results. And this is my last part of the presentation, and I will move away from nanotoxicology, but show you how I think we need to improve our models, or how we can improve our models to have more realistic output. And I think, and I will try to show you a short movie. There's one, well, there are several groups also in Europe, but there's one group in the US that is really dominant and doing very nice things. It's always risky to show a movie, but it seems to work after some commercials. And they have some nice models developed for lungs, but also for the intestine. This is an example of a lung. So it's the Wyss Institute at Harvard. They make devices where they have membranes in the middle and two vacuum chambers along that. And now you can grow on this membrane, which is not different from a Transwell or traditional well. You can grow cells, in this case lung, but they also have used compatible models for the intestine. You can grow cells on top of it and on the bottom side of it, endothelial cells, for example. And by playing with the pressure, you can stretch your membrane. And the nice thing, and Barbara also hinted on this, that you can, of course, in this case have an air channel, but in a gut conditions, you will simply have a lumen condition where you flush the intestinal content through it, and on the other side, you have your plasma side. Well, and then this is part of a TED talk. It continues, and it in the end shows also some very nice intestinal things, but I will stop it now here. Stop it. And go back to my presentation. Oh, that's nice. So I will start again. Or is it still here? Here it is, sorry. So this is, I think, they paved way for all kind of microfluidic models. We are using a little bit of simpler model. It's illustrated here in cartoons, where you have still a static membrane, but a flow on top and a flow below that. Grow our monoculture or co-cultures on this membrane and flush compounds through it. The interesting discussion is also, can one use cell lines for this? The American groups claim that these Kako cells differentiate in all kinds of cells that we have in our intestine, or should one use stem cells? In the Netherlands, I think the Hubrak Lab is one of the most famous institutes that developed organoids from human stem cells. So I guess I need to start very soon with trying to incorporate these organoids also in these kind of models, simply to again increase the biological relevance of these models. This is the current experimental setup we have in the lab, where we have various chips. These are, let's stay here perhaps. The chips, as I had in the cartoon, are as small as a microscope glass. And we have now systems where we have 4 of these chips in one holder, so that we can use parallel exposures and vary the conditions very easy. And what you will get then is, well, first I think we need to establish this model and see if it's really changing the model. Is it improving the model from a biological perspective? So we compared in an initial study how do these cells mature and compared the Transwells to the chip, and simply performed classical stainings of the nuclei, which are in blue again, the tight junctions, and the cytoskeleton, which is in green. And the cytoskeleton, I think, is especially interesting because that would show the polarization of the cells and the differentiation, maturation of the cells. This is the condition we have in Transwells. Also here, perhaps not always as easily seen on the screen. It's much better, I see from here. traditionally, and also what's claimed by the colleagues from the WIS Institute, is that in Transwell you have a very flat cells, not really a 3D structure. What you see here is a little bit, I would say a little bit of a 3D structure, but what's sure that it takes quite a long time. Traditionally, it takes about 21 days before you have a well-differentiated cell system with polarized cells which are tight, so that the tiers values are high enough. And if we use molecular markers to study the uptake or the absence of the uptake, the tightness of the model, it's debatable, but usually we have to wait for 21 days. Some people wait a little bit longer. But if you now compare this to the chips, and this is, please realize this is only with chips with only flow, so not with extraction. In a much shorter time, here the example is 7 days, we have a more prominent 3D structure. I think I'm always a little bit careful. So is this now really a village structure, or is this a hill in the architecture? But for sure it's more of a 3D structure than in the Transwell. And if you again look at these kind of images, this is focusing only on one of these structures. I think you really see that there is a more complex structure being formed. We did not expect it as drastically as this one in our initial experiments. So there's a glass on top of it, so it really pushes down the 3D structure a little bit. But it's showing and indicating that the shear force, so the liquid flow in these models are important for the 3D structure of this kind of models. So subsequently we exposed our systems to dioxins. As I indicated, I'm working mainly in the Institute of Food Safety. Dioxins are of course the compounds that we find back in the environment, but also in food. So the question was, if you would compare the traditional Transwell models with the chips, would you then see a different uptake of these dioxins? And dioxins is not one compound, but we have many congeners, PCB-like dioxins and just normal dioxins. And we compared the uptake and translocation of the different congeners of the dioxins. And what's striking, I think, is that the pattern of translocation of these compounds is not different in the Transwells compared to the chips. But if you look at the axis, the amount of dioxins that translocate in these systems is really different. And then the nasty question comes, and I will address it myself before you will ask it perhaps, which of the models is now of course predicting the human situation better? And this is a difficult question. We need to go more in the literature and try to find either human data in dioxin plasma levels, or rely again on animal studies, which I'm not a favorite of, because how adequately do the animals now resemble the human conditions? This is about it, but there's more in these kind of models. What I've not discussed so far is the microbial presence in these models, and of course we all know that the microbial community, especially in our intestine, is very prominent. In the last decade, I think we learned that the microbial system is shaping us. At least it's shaping our immune system. And it's also clear that in toxicology that food ingredients or toxicants are metabolized by our microbiome. So if you want to have a realistic model, I think you need to try to incorporate a relevant, realistic microbial system there. Again, the American people have a very nice publication in PNAS where they use this their cut on the chip models with really simplified by microbial systems. And I just last week submitted a proposal to try to do that a little bit better. Together with the microbiologists at our university, we will try to simulate a representative fraction of our microbial system that is responsible for metabolic pathways, to grow them in the lab, in our models, and this is again a cartoon of course, grow them under anaerobic conditions, which is a challenge for the engineers to make a chip and have on one compartment anaerobic, so without oxygen compartment, and on the bottom side, because we will have cells there, they need to survive there in the presence of oxygen. Grow either cell lines or more advanced stem cell based intestinal systems. Co-culture them with the local inflammatory system to really study the interaction of the microbiome and the host in relation to nanoparticles. Then we would, I think, do really sexy research, or just food ingredients and study the triangle between the host and the microbe interaction. That can very elegantly be done, I think, in microfluidic models. What also is very interesting is then to incorporate, because this is not only biology, it's also highly technical. And the engineers would love to have all kind of chip technology in there, either to simply measure the resistance and to show that the cells are tight, so again measures of the tier values, but easily measures of oxygen, pH, or if you incorporate, and that's all kind of these things are possible, antibodies or SPR-like technologies, you can online in your system measure the compounds of interest, cytokine expression of your cells, or the translocation of compounds. Near future, because we have a PhD student that will start on this, and far future, because well, there's plenty of things here to develop and to support the study of the biology in our intestine. This is it. I would like to thank my colleagues, both on the nanoparticles, nanodetection, and a combination with the recent work on the microfluidic models, and of course the funders that made all this research possible. Thank you.
Thank you, Hans. I think we have time for one quick question, if somebody has a question for Hans.
So in the workshop that Barbara referred to when we were talking about the lung on the chip model, the MIT or Harvard group that you referred to have this lung on a chip model with a flexing membrane to represent the lung. But the problem was that when they wanted to do any molecular endpoints, they had to harvest and pool cells from so many chips to get enough material to be able to do their analysis that it actually became very expensive. So have you found any ways around that will help you with the lung on the gut on the chip to be able to do the analyses that you need? Do you have enough cells there to do it?
In our current system, the amount of cells is comparable to what we have in Transwells. So we have simply, I guess, a larger surface where we grow the cells on. So this could be a very pragmatic solution.
I think the MIT model's gone too small too quickly there, way ahead of the analytical.
Yeah. And the other thing would be to, I think, use advanced analytical methods to really measure these compounds. What we aim to do is open the chip and use laser ablation MS to really generate spectra of the metabolism in the cells. So it's stepping us away from the molecular biology kits to more advanced detection. Then I want